Hello guys, and welcome to our video on the cell membrane. Today we're going to be discussing three major themes: the fluid mosaic model of the cell membrane, selective permeability, and transport of various solutes. So let's start off with our first theme, the fluid mosaic model. So every cell has a plasma membrane, as we know from our last video, and the plasma membrane's job is to primarily to protect the cell from its environment, but it also has various secondary jobs such as selecting which solutes should enter and which solutes should not enter, as well as communicating with other cells. So these plasma membranes are composed of lipid bilayers, and so lipid bilayers are organized in a way where the hydrophilic ends are located facing the interior and the exterior of the cell, and the hydrophobic tails are located so that they face each other to prevent themselves from touching the water. And so when we have molecules like these lipids, we call them amphipathic. Amphipathic means having a hydrophobic end and a hydrophilic end. And so just like there are amphipathic lipids that make up a lipid bilayer, there are also amphipathic proteins, which are also scattered throughout the membrane, and they have various functions, as you'll find out later. So to keep the cell membrane from freezing or liquefying at different temperatures, there are various mechanisms in place. So for example, unsaturated hydrocarbon tails can help prevent solidification of the membrane at cool temperatures. So as you know from our last video, when you have unsaturated hydrocarbon tails, that means that the hydrogens are not closely packed together, and so when they're distantly placed, that prevents solidification. And cholesterol, which is often talked about from a nutritional standpoint, also prevents solidification of the membrane, but can also prevent liquefaction of the membrane at high temperatures. So cholesterol helps keep the cell membrane stable, in other words. So we also have glycolipids and glycoproteins, and these are found on the exterior of the plasma membrane, and so they serve key roles in communication and interaction with other cells. So for example, these glycoproteins and glycolipids are key to communication. That, for example, there may be a pathogen in the organism, and certain cells communicate with each other using these glycoproteins and glycolipids to indicate that there is such a pathogen, and so that can activate other cells which serve to exterminate the pathogen. Now let's talk about our second theme, which is selective permeability. So cells are selectively permeable. That means that they are able to allow certain molecules to pass through. So they're not like a brick wall, which you can't really move anything through without breaking the brick wall. So it's not like a brick wall. So it can allow molecules to pass through. However, it doesn't allow all molecules to pass through. Because if it allowed all molecules to pass through, then the purpose of the cell membrane would essentially be defeated. So it doesn't allow that. So hydrophobic substances tend to pass through easily because the interior of the cell membrane is hydrophobic due to the fact that the hydrophobic tails are all pointing towards each other, so that makes that area hydrophobic. But certain other substances that are crucial to cells, such as various ions and water, those require transport proteins because they do not pass through hydrophobic areas as well, because they tend to be hydrophilic. Aquaporins tend to increase the permeability of water through the cell membrane, and they can be really, really important, especially in the kidney, or they save massive amounts of water from being removed from the body without reason. So aquaporins are really important proteins. Now let's talk about our third theme, which is transport. So diffusion is essentially the movement of a molecule down its concentration gradient. So concentration gradient is kind of like the direction from which there's more to less. So let me show you a good example of this. So as you can see in our first diagram, molecules of dye, the concentration gradient is going from left to right because there's a lot of this yellow molecule on the left side of the diagram, whereas there's very little, or in this case none, on the right side of the diagram. So net diffusion is also in the same direction as a concentration gradient, and this is what we like to call downhill movement, because as kind of like rolling a rock downhill, this doesn't require any active use of energy. So as you can see over time, since these molecules are moving rather randomly, each of them individually will move randomly, but over time they will gradually move towards the right side until, of course, both sides are balanced, and this balance is called equilibrium. So that's essentially what diffusion is. So osmosis is the diffusion of water across selectively permeable membrane. Oftentimes this brings up lots of confusion, but osmosis is simply diffusion of water. So for example, let's say like outside a cell, 97% of what's outside a cell, the solution outside of a cell, is solute, and the other 3% is water. Let's just assume that, okay? And then let's assume that on the interior of a cell, 95% is solute, and the other 5% is water. So what would happen in this case? Well, in this case, the interior of the cell would gradually release water to the exterior of the cell, because the concentration inside is 5%, concentration of water, that is, whereas outside is 3%. So they're going to keep moving until both sides become 4%. So that's a good example of osmosis. However, when there are multiple solutes, each is going to move down its own concentration gradient, and it will be unaffected by the concentration gradient of other substances. So let's take an example. In this second example, B, diffusion of two solutes right here, we can see that there's a yellow substance and a purple substance. And so each of these substances, as you can see, will move independently of each other until each reaches its own equilibrium. So that's a really important idea to keep in mind, is that the diffusion will occur independent of the diffusion of other solutes. So let's also cover tonicity while we're at this, because tonicity is the amount of solutes in a solution relative to the amount of solutes inside the cell. So let's say that a solution is hypertonic. That means that it has more solutes than the organism. And so if in the absence of other adaptations, the organism will shrivel because the water will leave by osmosis when the solution is hypertonic. Now, plant cells will fare best at hypotonic solutions, but most cells will not. Instead, most cells will just blow up because the water will gradually seep in, and in the absence of other adaptations, that will just result in the cell eventually blowing up. However, plant cells have a cell wall, and that helps create what is known as turgidity, I believe, and that helps keep the plant cell firm and doesn't make it explode as would happen in various other cells. However, animal cells fare best in isotonic solutions. So in isotonic solutions, that means that the concentration of water outside the cell, or the concentration of solutes outside of the cell, and the concentration inside the cell would be the exact same. Now this is a really strict constraint. So many organisms have developed adaptations for survival in conditions that aren't favorable to those cells. So a good example would be a species called Paramecium caudatum, and in that species, it lives in solutions where naturally water would continue to seep in into its body, so it lives in a hypotonic solution. However, to survive, what it does is it has a contractile vacuole, and this contractile vacuole, what it does is it gradually pushes out the water as it comes in at an approximately equal rate. So the net result would be that there's no effect on the protist, or that organism. So as you can see many examples throughout nature, various plants, as the example of the cell wall proved, and animals and various other organisms have such adaptations. Now let's move on to our next topic. So that is, since the speed of a molecule diffuses, is proportional to the square of the distance to be diffused across. So that just means that if there's a relatively small distance, then most of the time diffusion will occur at a relatively low won't take a lot of time. But if there's a massive distance, that means that it would take massive amounts of time for a solute to diffuse across. So for this reason, many molecules have associated transport proteins to speed up the movement of those molecules across plasma membranes. And these evolved because in organisms that lack these transport proteins, they probably died because crucial molecules such as water or sucrose or other molecules would take massive amounts of time to move across, and that wouldn't be fast enough because the cells are constantly using up energy through metabolism and they need replacements, otherwise they will die. So over the course of evolution, those organisms with transport proteins tended to survive more, and so that's why most organisms today have transport proteins to speed up the movement of those molecules. Now, all we've covered so far is primarily concerned with passive transport, which does not require any input of energy. However, active transport is a form of transport where ATP has to be used to move a molecule up its concentration gradient. So in other words, ATP is used to move in a molecule from where there's less of a molecule to where there is more of a molecule. So for example, if a cell needs lots of a certain molecule, then probably outside the cell there will be very little of that molecule remaining, and there will be massive amounts within the cell. And so what the cell will do to remove the remainder within it is it's going to use ATP to move those molecules up that gradient. Co-transport is a form where passive and active transport are essentially combined. So a certain membrane protein is used to use the downhill diffusion of a solute to provide energy for the transport of another molecule uphill. So here's a good example of that. This is what is called a sucrose hydrogen ion co-transport. So as you can see right here, the active transport is going on with this proton pump because ATP is being used to expel these hydrogen ions. And so the plus signs here indicate where there's more of a certain molecule, and the negative signs indicate where there's less of a certain molecule. So obviously the H plus will not diffuse across the cell membrane without the use of ATP. However, since that is being used, the hydrogen ions gradually will be pumped out, and then they will diffuse back in because there's way more of these hydrogen ions on the outside than on the inside. And so as these diffuse back in, energy is being given off. Why? Because of the law of conservation of energy, which states that energy cannot be created nor destroyed. And so since this hydrogen ion is going from a high energy level down to a lower energy level, that energy has to be given off somewhere. So that energy is essentially absorbed by the cell, and the cell can then use it to move certain molecules from where there's very little of them to where there's more of them. So in this case, where there's more of them would be within the cell. So for example, it uses the diffusion of the hydrogen ions to move sucrose from into the cell. Since a cell needs lots of sucrose, this whole sucrose hydrogen ion co-transport is necessary. So I hope you enjoyed our video on the cell membrane, where we covered 3 major topics: the fluid mosaic model, selective permeability, and transport. Subscribe and like.